Hello. I hope you guys are doing good. We're here to talk about chapter 15, Microbial Mechanisms of Pathogenicity. I love this chapter. I think it's really interesting because this chapter are the tools that we're going to use in the following chapters when we start to talk about infectious diseases to understand how each bacterium causes disease. So when we think about how one bacterium acts, what type of toxin it produces, how it gets in the host through which path, and how it attaches to the host, and what kind of damages it causes, it's all what we're going to talk about in this chapter. So we're always going to have to come back to chapter 15 to recall what is causing the damage and how the bacterium causes disease or virus or another microbe. We're mostly going to talk about bacterial mechanisms, and then briefly we talk about viral, protozoa, and helminth mechanisms. So when you look at the learning objectives here, this is what we're going to be talking about in the first video about this first part, and then in the second video about the second part, the second list here. So now we're talking about portals of entry. So we talk about how they enter the body, how they attach, which enzymes they produce to cause harm, what kind of mechanisms they use to fool the immune system and survive inside the host, and then how they really damage the bacteria, the host cell. I'm talking about bacteria here, right? And then in the second video, we talk about toxins, and then we talk about a little bit about viral, fungi, protozoa, and helminth pathogenicity. I made this table for you. And this table, it doesn't need to be turned in. It's just something, a tool for you to use as you were talking about, as you were studying about all these mechanisms and molecules. One thing you could do is like divide this into two parts, in two papers, so you have space to like fill them up, or just print the way it is and start filling them, because I think you can kind of summarize everything you need to know about this chapter here. Let's say 90% of what you need to know, okay? So what is pathogenicity? Ability to cause disease. And then pathogenicity is measured by the virulence, which is the degree of pathogenicity. How harmful a microbe is to the host is the virulence. And the virulence is going to be defined by different things that the microbe can do to achieve it. So let's start with how they enter our body. So they can enter our body through mucous membranes, skin, or parenteral routes. Let's say, what are the mucous membranes, where are they, and how that's so important? Actually, mucous membrane is the main port of entry, okay? Think about every hollow organ in your body. Remember A and P? Now we start to talk a little more about our body and A and P to understand the relationship between the microbe and host, right? So think about your respiratory system, hollow inside. Think about your urinary system, bladder, urethra, hollow inside. Think about your genitalia, inside the penis, inside the vagina, uterus, all the ducts from the male reproductive system, hollow, right? And now think about digestive system, mouth, esophagus, stomach, intestine, all hollow. So all this that we just mentioned have mucous membranes, which is the epithelia, right, inside, covering, lining these organs, these systems. Meaning, this is a very effective way that microbes use to attach to the membranes and enter the body, okay? The mucous membrane is wet It's sticky. They love it, right? The main one here that most of bacteria and even fungi use as a way to enter the host body is the respiratory system, okay? Now let's talk about skin. Can microbes enter intact skin, unbroken? They can. Hair follicle, sweat glands ducts. They can totally get through intact skin, through hair follicle, through the sweat gland ducts, and penetrate and get into the dermis, get into the blood, okay? What is keratin here? Keratin here, I'm just trying to tell you that there are some fungi that use keratin to cause disease. Why? Because they like to eat keratin. They like that protein, okay? So that's fungi that cause mycosis on the skin, usually like to eat the keratin. That's how they feed. Parenteral route. So if your skin is broken, cuts, or if you use an injection through the skin, if you get a bite from an animal or from an insect, all this, insects are animals, but like I was thinking about bigger animals or small animals, bites are ways through which microbes can enter your blood, right? Surgery. During a surgery, you can get some type of infection, right? So, and then microbes are deposited directly into the tissues when barriers are penetrated. Most pathogens have a preferred port of entry, but they can use different ports of entry. So now I want to tell you about a concept that is, think about this is the infectious dose for 50% of a sample population. What is the dose necessary for 50% of a group of people to get infected? Right? So that's ID50. So if you think that the ID50 is the number is high, that means you need a lot of particles to make you sick with that one specific microbe, okay? So I'm gonna give you an example. Bacillus anthracis, it can infect the skin with only, remember they form endospores, with only 10 to 50 endospores. If they're inhaled, then just a lot more need to be inhaled in order for Bacillus anthracis to cause respiratory anthrax. Okay? Ingestion, 250,000 to 1 million endospores. So now my question to you is, which portal of entry is the preferred one? Which portal of entry will make you sicker quickly with Bacillus anthracis? Pause the video and think. If you thought it is the skin, you are correct. So think as ID50 as being inversely proportional to virulence, okay? The less particles, the less bacteria, this is bacterial cell, right? If you think about virus, then we talk about virions or particles. The less cells you need to infect a person or any host, which is we're talking, the more virulent that microbe is, okay? Another example, since all we talk about in the world is the coronavirus. The ID50 for the coronavirus is here. This is actually, it's an older, we don't have this information really for, at least until now, for humans. So in 2010, there was somebody studying bats, and they, isolated the different types of coronavirus, including the SARS-CoV which is the one that infects baths. Actually, the one that is causing the pandemic is the SARS-CoV-2. And they were trying to see what is the infectious dose, the ID50, which is actually here, because this is ID10, it's 10% of the population. So to infect 50% of population, look at that. But this is in mice, okay? You only need 100 or maybe 200 particles of SARS-CoV to make an animal sick. That is 200 particles per ml of secretion, I think. So, which is PFU, which is plaque forming units. It's like how many each plaque that is developed into a tissue culture experiment is caused by one cell that was infected with one viral particle. So thinking that you need only 200 to infect an animal is a big number, right? So this is just to illustrate how this type of measurement is super important. When we talk about LD50, then we're looking at this, right? We're talking about lethal dose of a toxin, lethal dose of a toxin to be able to intoxicate and make somebody sick. Which toxins are we talking about? Toxins produced by bacteria. Examples: botulinum toxin. You need 0.03 nanograms per kilogram of the host to make somebody sick. What does botulinum toxin does? It causes a soft type of paralysis. Your muscles, we're gonna learn this in details, but your muscles don't respond to contraction anymore when you have infection with this toxin. So you have non-contraction type of events. Okay, so that's how it intoxicates you. So think about that, committing your diaphragm. You cannot breathe, right? So if it gets to that point, it's really, really serious. Shiga toxin. This is one of the toxins that is produced by Shigella and E. coli and causes diarrhea. 250 nanograms per kilogram. So you see, you need a lot more of Shiga toxin than a botulinum toxin to make you sick. Super important. Look, you need a lot more staphylococcal enterotoxin to give you diarrhea. But this enterotoxin is very common to cause food intoxication. So even though you need more, it's not that uncommon to see this toxin causing diarrhea in patients. Okay? Cool. Now we're going to talk about adherence. So almost all pathogens attach to host tissues in a process called adherence or adhesion. Adhesins or ligands on the pathogen, they bind to receptors. The main receptor that they bind here is called mannose. It's a sugar that is on the surface of the host cell, and the pathogen can bind to it because they have these adhesins, okay? These adhesins are usually present in the glycocalyx, in the fimbriae, and also microbes that form biofilms. They can share nutrients between each other, and that's a way to attach to. The biofilm helps the microbes attach to the host. So this is just to illustrate how this is a receptor like the mannose receptor, and then you have the adhesins on the surface of the pathogen causing that type of attachment. Capsules are also important in the initial phase of the infection, because remember, capsules are also glycocalyx and they're around the cell wall. But here we're more talking about they make the cell, it's kind of the opposite of adhesion here, because when microbes have capsule, they can have other adhesins to get attached to the cell, but the capsule is going to work like a wet soap, meaning phagocytes are going to try to catch the bacterium, but because the bacterium has capsule, they evade the immune system. They impair the phagocytosis. Examples: Streptococcus pneumoniae. So this one, Haemophilus influenza, and Neisseria are 3 microbes that have capsule that cause meningitis. We're going to talk about them later. Haemophilus influenza, strep pneumoniae, and Neisseria meningitis. Bacillus anthracis causes anthrax and has capsule. Yersinia pestis causes plague and also has capsule that makes them evade the phagocytic process by macrophages and neutrophils, for example, which are the main cells that perform phagocytosis. So now, still talking about external structures, right? We were talking about adhesins, capsule. Now we're going to talk about some cell wall components that are also important in attachment, okay? So there is a protein here that is called M protein that helps, that are present in fimbriae, and they make phagocytes resist. They make the microbe resist to the phagocytosis by the phagocyte. One example of a bacterium that has M protein is Streptococcus pyogenes. So M protein is all this. That's the Streptococcus cell. M protein is all this. And one thing that they do is that they inhibit they block IgA mediated phagocytosis. Okay, so then the phagocyte is here. The phagocyte is much bigger than the bacteria, but I'm not going to be able to draw this here really well. And the phagocyte has an IgA on the surface that was attached, that came from a lymphocyte, right? So this would be an IgA. And then this is going to impair the ability of the phagocyte to eat this bacterium, okay, because it blocks the IgA, okay, because that IgA would be specific for the microbe, and M protein kind of blocks that action, okay. OPA also helps with attachment. Neisseria gonorrhea, that causes gonorrhea, has OPA proteins. Wax lipids, it's another type of cell wall component, which is the mycolic acid, present in Mycobacterium tuberculosis and nocardia, that causes that type of wet soap kind of feeling to the cell. But even if the cell gets phagocytosed with mycolic acid stops the formation of the phagolysosome, which is the phagosome fusing with the lysosomes inside the cell, so the content can be digested. Meaning what? Mycobacterium gets phagocytosed, but because of the mycolic acid around the cell wall, the phagocyte cannot eat it, cannot digest the bacteria, it can kill it. And they live inside the phagocyte, like that's their little house, okay? Now we're going to talk about enzymes that are produced by microbes and are super important in causing disease. So we have coagulases. They help to coagulate fibrinogen. So coagulases do what? Staphylococcus produces coagulase. They are coagulase positive type of bacteria, and coagulase binds to fibrinogen and make it insoluble, transform it into fibrin without all the components from the coagulation cascade from the host. Kinases, they do the opposite. They digest fibrin clots. So the bacteria needs to move around, and sometimes there are clots being formed by the whole inflammation process, and there are some bacteria like Streptococcus pyogenes that produces streptokinase, which is a kinase specific for this bacteria that makes this digestion of fibrin clots. Hyaluronidase is an enzyme produced also by Streptococcus, among other bacteria, that is going to cause destruction of a hyaluronic acid. Hyaluronic acid is a component of the connective tissue, and connective tissue needs hyaluronic acid to make it firm, and when that's destroyed, that's how the bacteria is able to move around, because it's destroying the rigidity of the connective tissue and the host. Collagenase is important because it breaks down collagen. So it works similarly to the hyaluronidase, but in this case, you're not breaking down hyaluronic acid. You're breaking another component of the extracellular matrix, which is present in the connective tissue in a big amount. And the compound here is collagen. Clostridium is one bacterium that has collagenase. And bacteria like Neisseria gonorrhea are able to produce IgA proteases. So remember we talked about the M protein and how that would impair IgA action. So here, this enzyme breaks down IgA, okay? This is just a sad story, an example of how streptokinase from Streptococcus pyogenes or group A Streptococcus acts. So streptokinase is going to act on the plasminogen, and that acts on the plasmin, and is going to break down fibrin that is already forming a clot. So by breaking down clots, look what can happen. This is necrotizing fasciitis caused by Streptococcus pyogenes, and because clots are not being formed, the bacteria is being able to move through tissues and through the blood and cause all this harm. There's also a lot of cytokine overproduction here that makes this look even worse. And one thing that is also important about this type of lesion, where you see these darkened areas, is this is the effect of hyaluronidase. Okay, the fact that hyaluronidase is breaking down hyaluronic acid is what causes this type of lesion that has these blackened areas. You see this also when Clostridium perfringens causes damage in skin in gas gangrene. That's very typical also of that type of lesion. All right, so now we are going to talk about antigenic variation. So we think about this as the antigens trying to do something to fool the immune system, right? So for example, the influenza virus can change some molecules that are the spikes that are present in the envelope of the virus to fool the immune system, because we learn to attack one specific type of molecule and then they change to a slightly different one, and then our immune system cannot recognize it. Same thing for Trypanosoma brucei, which causes African sleeping sickness. If you think about this disease, this protozoa is able to change antigens that are also recognized by our immune system every week. So during somebody's disease, every week you have a different clone of Trypanosoma. So the immune system can never catch up with all these differences. When we think about bacteria, always remember the C, the H, and the O antigens. So these are all external antigens of bacteria. And you have C is for capsule. And then you have the H, which is for flagella. So antigens in the capsule, in the flagella, or in the cell wall. will change to fool the immune system and evade the immune response. Invasins are molecules that some microbes use to help them penetrate the host cells. So they use the host cytoskeleton to, so the membrane of the host cell makes what is called membrane ruffling. And membrane ruffling is just a projection of the cytoskeleton towards where the bacteria is, manipulated by the bacteria. But this is happening in the host cell. So if you look at this picture, these are the rufflings here. See? And these are the bacteria digging inside these membrane rufflings. Okay? You have Shigella and Listeria. Also, so this is actually common in Salmonella and E. coli. But you have a different type of manipulation of the immune system that is similar to this, but in this case is more for Shigella and Listeria being able to move from cell to cell. Listeria usually infects phagocytes, and then it can jump from one phagocyte to the next because it induces one phagocyte to project pseudopods so they can reach that part of the cell and get into it. Okay? They're smart. This is another now how they use another type of attack from bacteria. How they use the host nutrients in their favors. So there is a molecule that is called siderophores, and siderophores are proteins that are secreted by the pathogens that are able to sequester iron. So this sequestration of iron happens when the cell is producing iron, because it's going to use to have a host needs the iron for a good effect, good performance of the immune system. Also is needed for oxidative burst in bacteria and in phagocytes when they phagocyte the bacteria. So the phagocyte uses reactive oxygen species inside the phagosome to be able to kill the bacteria. Iron is needed for that to happen, and then you don't have that specific type of response in the phagocytes because the iron was sequestered by the bacteria. And also, all the bacteria, all the microbes can cause that are pathogenic, can cause disruption of the host cell function. So this is a direct damage, right? They can use the cell's nutrient, like the siderophore, like I just told you, production of wastes, and multiplication inside the host cell, causing the cell to rupture. So next video we start talking about the toxins. I will talk about this to you soon.